So I'm Nathan Skene. I'm a group leader in the Dementia Research Institute at Imperial College. So we do our work using genetics and single-cell genomic data to understand basic questions, just simple questions about what causes brain diseases. As it was commonly assumed until a couple of years back that Alzheimer's was a disease affecting neurons. So it's a brain disease. It causes degeneration of neurons, it causes loss of memory, and so the default assumption was it was a neuronal disease. But what we can establish now from using genetics is that the driving factors, on a genetic level at least, which cause the disease, don't act via neurons. So we can infer from this that Alzheimer's is in fact it's more of an immune disease. There's a type of cell in the brain called microglia that are the immune cell of the brain. And that appears to be the focal point for Alzheimer's. The majority of genes actually are quite specific in where they are expressed. They're tailored towards the function of particular parts of the body. And so we take advantage of that. Because they are specific to certain parts of the body, if lots of the genes involved in a particular trait or a particular disease have a level of specificity to a particular part of the body, then we infer that that part of the body is involved in the trait. To understand where genes are expressed, we use a technique called single-cell genomics, or single-cell RNA sequencing. If you take a piece of tissue, say a piece of brain tissue, then what you kind of have there is a fruit smoothie. You know that it used to be comprised of strawberries, bananas, raspberries, but you've now just blended it all into one thing. You can't distinguish anymore what went into it. You just have an average profile at the end, the smoothie. And with the brain, we know that there's a huge number of different cells go into a piece of tissue. We established a couple of years back that there's probably at least 40 different types of just interneurons within one small region of the brain. So that's the kind of complexity we're dealing with. And so what we do now is we take the tissue, and instead of making the smoothie, we break it down into every single individual cell. We separate it off from the cells that surround it, and then we analyze it on its own. So what we do is we sequence every piece of RNA found within each individual cell in such a way that we've barcoded each chemical reaction that occurs. We know every molecule that was in there because we put a unique tag on it that told us that it was this molecule from this one cell.